Okay, all right. Okay, so we're going to begin with biochemistry and the molecular portion of biochemistry, that is genetics. That is exactly what we're going to begin with. Really excited over here. Nothing to be worried about. Easy stuff. I'm just going to tell you exactly what's going on over here, how you are supposed to face these questions in the step one, and in no time we will be done with this very soon. Having said that, we will come across certain pathways. Okay, we will come across certain pathways, like we will come across de novo, pyrimidine, and purine pathways, and then purine salvage pathways. So for these pathways, what I need you to do is, when I would be drawing these pathways on a blank page, I need you to have a notebook or a piece of paper ready on the side. So when I am drawing something, I need you to draw the same thing. Okay, so what that would do is, if you draw the same thing which I am drawing on the blank page, at the end of the day, you can keep those notes. Okay, you can keep those notes, and either you can post it on your wall or you can keep it on your notebook, and that would allow you to keep all the pathways in one book. So if you have to do a quick revision and recapitulation, most of the questions are usually from the pathways. Most of the questions are usually from the pathways of the genetics and biochemistry. Very handful amount of questions are expected from the text. We're going to tell you what are those questions. But when we are doing the pathways, please make sure that you make your own notes for the pathways. Keep a notebook. Okay, when we are writing something, write it down so that when you want to do a quick revision and recapitulation, you can go through the pathway book which you have created. Okay, have I been clear? Have everybody understood what I just said? Okay, good, very good. Okay, so without further ado, let's begin today's lecture. First of all, the first thing that we're going to study today is the structure of chromatin. And before we jump into this, let's do a small introduction to genetics, and that's that. Genetics is basically the study of your genome and all the structures that are in it, right? So if this is basically a cell, right, and then this is the nucleus, then the nucleus is nucleolus, and then over there in the nucleolus we have what we say is the genetic structure. We have the DNA, all right? We have the DNA, right? So DNA is basically deoxyribonucleic acid. It's a nucleic acid over there in the DNA. In the DNA, okay, in the DNA, what you have is you have small pieces of informations. You have very small pieces of informations, right? You have small pieces of informations in um, in the form of in the form of what we say is chromosomes or chromatins, right? And those chromosomes, they contain, they are filled with even minor informations. Okay, they are filled with even minor informations that are known as genes, right? That are known as genes. And in those genes, okay, and in those genes, you have each and every information at each and every position. So first of all, you have the DNA, and the DNA, okay, you have the chromosomes, right? Chromosomes have DNA, and those DNAs, you have genes, right? And those genes are located, these genes, each and every gene can be categorized into alleles. Those alleles are located at a single position of the gene, right? And those positions are known as locus. These are some minor terms which we have to get accustomed with. Have we understood this? Have we understood this small piece of information before we even begin? We have chromosomes, right? How many pairs of chromosomes do we have? Chromosomes. How many pairs of chromosomes do we have? We have 23 pairs of chromosomes. That's correct. Each and every chromosome it contains the DNA structures. Those DNAs, they have small pieces of information that are called genes. Those genes are located. Each and every gene is known as an allele, which is located at a different position. The positions are known as locus. That's that. So right now we're going to study of how a chromatid or a single chromosome is formed from the gene. Uh, from the gene, meaning that how the DNA will structure itself to form a chromosome. That's exactly what we are going to study, how that is happening. And the reason why this is important is because clinically, we can use the activation or deactivation of chromosomes for different purposes. Okay, so clinically, it's important. That's why we have to study this. First of all, the structure of the chromatin. Chromatin is basically DNA. It exists in the condensed chromatin form to fit into the nucleus. So DNA is basically condensed because DNA is such a long piece of information. If it was not condensed into a smaller piece, it would not be able to fit inside the nucleus. So the DNA is condensed to form a small chromatin form to fit inside the nucleus. The DNA, as you can see over here, it loops twice around a histone. Histone is basically, it's nothing but a protein. Okay, so histone, it it loops twice. So the DNA is condensing. After it condenses, the DNA, it loops twice around the histone. And after it loops around the histone, it forms what we call is a nucleosome. Nucleosome are basically these structures over here. These structure meaning that this beads on a string appearance, these beads on a string appearance with each and every histone containing two loops of the DNA. This is known as a nucleosome. Okay, then there are different types of histones. You have H1, H2A, H2B, H3, H4, right? So each and every one is a histone over here. H1, it binds to the nucleosome and to a linker DNA and thereby it stabilizes the chromatin fiber. So basically, the H1 will bind to the nucleosome and it will bind to the linker DNA. It will bind to one nucleosome and then it will bind to the another portion of the DNA, and then it will stabilize the chromatin fiber over here. Till now, from this paragraph, you have no questions which are coming. Okay, no questions will be asked from this paragraph. And this is just for your understanding, that's all. Okay, next one. Next one is the DNA structure has a negative charge and a positive charge. Okay, the DNA has a negative charge and a positive charge. The phosphate group, okay, so what I need you to do is I need you to write this over here, DNA, okay, DNA. Negative charge is from the phosphate, okay, negative charge is from the phosphate group. This is an NBME question. This is a question from the NBME. Okay, the phosphate group, it gives the DNA a negative charge. Lysine and arginine gives the histone a positive, histones a positive charge. So you have the DNA, which is the negative, which by the help of the ion, that is phosphate, and histones, they have a positive charge. So write down over here that histones, they have a positive charge. Positive charge. Why? Lysine and arginine. Okay, lysine and arginine. So write this down in your book. These are NBME questions. Okay, questions from the NBME. That's that. Okay, so lysine and arginine are basically, they are amino acids. Okay, they have a positive charge, and the DNA, they're given a negative charge by the phosphate, and that's that. In mitosis, the DNA will condense to form a chromosome, and the DNA in histone synthesis, it occurs during the S phase. This is also a question. This is a question. At which phase of the DNA cycle does DNA, at which phase of the mitotic cycle does the DNA in histone synthesis occurs? And it's at the S phase. And we have talked about this before. that in the S phase, you have the DNA which condenses, and then you have the structures which are getting ready to separate, right? So at the S phase, the DNA and the histone will the DNA and histone synthesis will occur at the S phase. Okay, next one. Next one is the mitochondria. They have their own DNA, which is circular, and it does not utilize the histone. The fact that mitochondria has a circular DNA, give this 5 stars. Okay. Next time you receive a question on NBME that which organelle has a circular DNA, your answer is mitochondria. Circular DNA is mitochondria. So mitochondria has a circular DNA and it does not utilize histones, meaning that the DNA in the mitochondria, it does not need the histones to compact the structure into a chromatin form. It can just compact by itself because the DNA is a circular DNA. Okay. So far, have we understood all the questions which will be received from this paragraph? Have we understood all the questions that will be received from this paragraph? Okay. 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 So DNA, the positive charge of the histones, which amino acids are responsible for the positive charge of the histone? Which amino acids are responsible for the positive charge? Yes, very good. It's lysine and arginine. Which ion is responsible for the negative charge of the DNA? Okay, good. Very good. Then DNA synthesis occurs at which phase? DNA synthesis and condensing. Good. Which organelle has a circular DNA and does not contain any histone? How many loops does the DNA make around a histone to form a nucleosome? Okay. Okay, good. Very good. Okay. All right. So we are going to discuss this diagram after the end of this page because we will study these things first and then we will understand what they're talking about. After that, we will jump into this diagram. Next one. Next one is heterochromatin. What is heterochromatin and euchromatin? These are two terms which we have to understand. Okay. Heterochromatin is basically the condensed DNA. DNA, when this is very condensed, it appears darker on the electron microscope. As you can see that over here, it is labeled H. This H is the condensed DNA. This extremely condensed DNA is known as heterochromatin. And it's sterically inaccessible, meaning that this heterochromatin is basically inactive, meaning that if you try to if the genes in the DNA they decide to form a protein, okay, for example, go through the process of transcription and translation and then form a protein structure, this will not happen because heterochromatin are inaccessible. So this is also another piece of information, okay, that is transcriptionally inactive. Transcriptionally inactive is heterochromatin. Heterochromatin is transcriptionally inactive. And transcriptionally active is euchromatin. Transcriptionally active is euchromatin. Okay. So basically, this is transcriptionally inactive, meaning that for a DNA to be transcriptionally inactive, one thing has to go up, another thing has to go down. Always remember this: when a single portion or when a certain portion of the DNA is methylated, when something is methylated in the DNA, it is inactive. When something is acetylated, it is active. So if heterochromatin is inactive, then there should obviously be increased methylation and decreased acetylation. Okay, decreased acetylation. The easiest way you can remember this is M for mute, and A for active. A for active. So next time, if you receive a question in NBME where they are talking about acetylation, try to